and thank you, Rafael, for the invitation and for being able to participate in this discussion. So, as the title indicates, I will be talking about regulatory networks in the context of evolution and development, in particular in plants. Okay. So, when we look at this diversity of forms and shapes, we would like to know how these shapes are generated and how they change, in two different timescales. First, in the scale.
How do you choose your transparency paper? Dish or
Sorry?
Is that a dish, food, or?
No, these are seeds. These are all seeds. All seeds. From trees in a Panama forest. So, we would like to know what they all have in common, structurally, for example, but also what are the differences between these seeds, and how these seeds change in two different timescales. In the timescale of development, which consists in the transit of a single cell to a structured and complex organism with organs and structures, and also in the evolutionary scale, which is the scale of many generations and in very long periods of time. So as Rafael was saying in his talk, we biologists usually are overwhelmed by this diversity, while physicists think what these things have in common and find them actually very similar. I think it's very interesting to study both aspects of shapes and forms in organisms. It's very interesting for us to understand which are the common features of all these structures, but also how these structures can vary within individuals, but also in populations, and eventually giving rise to different populations, to different species or lineages. So, more specifically, this question here can be put into three more specific questions. So, how the interactions among molecular, cellular, organismic, environmental, and other types of factors give rise to shapes and patterns during development. And with development, I mean to this transit from one single cell to a complex and a structured organism. How the changes in development of organisms can generate evolutionary innovations. This is very important to us, and this question in particular has given rise to a whole new area of study, which is usually known as evolutionary developmental biology, or EVODIBO. Well, it's not so new now, but it has been around for some decades. But there are still many central and interesting questions open in this area. And, well, another question that we would like to address is what are the common and particular aspects of patterning and morphogenesis in different lineages, for example, in these structures. Well, in order to address, or at least try to address these questions, we have chosen plants, and in particular Arabidopsis thaliana, as model systems. Why? Well, first of all, they have a very intense post-embryonary development. So in contrast to animals, plants continue to generate new structures, flowers, branches, leaves, etc. throughout their whole lives. And only some animals can do this, and only to some extent. So this is a very interesting aspect of plant development, and also their development is very plastic. This generation of new organs and structures responds largely to environmental factors, which makes their development very sensitive to the conditions in which plants develop. So they are a great system to study the interactions between organism and environment, not only in development, but also in the evolution of shapes and forms. Also, not lost, but lots, there are many data for model plants, such as Arabidopsis thaliana, although they remain largely disintegrated. So we think that also an important challenge in plant biology at the moment is to try to integrate some of the very detailed and careful and thorough data that have been generated through decades. Finally, since most studies in evolutionary developmental biology have been done in animals, we think that it's also very important to try to study development and evolution in plants, so that we are able to perform broad comparative studies between these two major multicellular groups. So if we want to know if there are common or generic aspects of development in multicellulars, then in multicellular organisms, then it's very useful to have also examples beyond animals, and plants are a very good model to do this. So we have been using models to try to address some of these questions in plants. And the idea of the models we are using is, first of all, maybe not the most important, but it's also a very important aspect of the models we want to use, is that they provide a framework to integrate all this data I was talking about. So we have, in many systems in plant biology, a lot of data of good quality, but we now need to integrate them into models that help us understand how plants actually develop and change shapes and patterns and so on. So models are a very useful tool to put all this in a common language, let's say. Also, these models, the models that we want to use, allow us to study the collective behavior of molecular and environmental and organismal elements that all interact during plant development. They also, as you all know, provide a tool to perform in silico explorations and experiments that will be very difficult to perform in the lab or in the field even. And we would like also to generate new predictions that can feed back experimental work, so that joint experimental and theoretical efforts can move forward together. Okay, so one of the central questions in developmental biology, and here from now I will be focusing on plants, is how cells with the same genome differentiate into cells that look as different as these ones. These are all cells from Arabidopsis thaliana. And well, part of the answer is that all these cells are subjected to different stimuli, for example, gradients of some orange morphogen. And then depending on their position, they can differentiate or acquire a different cell type and then differentiate. However, this model has some limitations, in the sense that, well, now we have to explain how these gradients are created, right? And another limitation is that this model considers cells as passive agents that somehow read these morphogens and then act or differentiate accordingly. However, what we have seen in plants is that cells that are being differentiated communicate with each other and with their medium, and they create these gradients in the same process of being differentiated. So there is not such a clear difference between these fields and the readout and then the cells, but rather it's the cells themselves and in the tissue in which they are embedded that are creating these gradients and fields that in turn can specify different regions of cell identities. So we want to know how these patterns of proteins, morphogens, hormones, etc. emerge during cell differentiation. As Tomás was explaining a few minutes ago, another part of the answer to these questions of how cells with the same genome can acquire these different identities is that, well, although they have the same genome, they have different sets of genes being expressed or active and inactive. And this gives rise to different forms and identities of the cells. The set of genes that are being expressed depends on the regulatory interactions of these genes with other genes, just like Tomás was explaining. So I won't stop on this very much, but genes can produce proteins that activate or inhibit the expression of other genes. And this can be rather naturally translated into networks of positive and negative interactions. So we have been using the network formalism to study gene regulation and in turn to study cell trait differentiation in plants. So just to explain very briefly the type of model we have been using, I will explain this, let's say, toy model, in which we have 3 genes, X, Y, and Z, that sustain positive and negative interactions among them. And since this is a very small system, and the variables have discrete values, so they can only be in the value 0, which is inactive, and 1, which is active, we can specify all the possible conditions of the system, and based on our knowledge of the system, experimental data, and so on, we can specify a function, in this case in tables, that tell us what the state of the system will be in a time unit after. No? So this is the system at time t, and this is the system at time t plus 1. Something interesting is that all these discrete systems eventually attain states that are called attractors that can sustain their own configuration of zeros and ones. This is why they are called attractors. And these attractors were thought by Stuart Kauffman in the late 60s to represent cell types. And so he thought that the number of cell types that an organism can attain depends on the architecture of these types of regulatory networks, and then in the number and nature of the attractors that these networks have. As you can see, these are very qualitative models and are not useful to address questions such as what concentration of certain protein we will find at certain time in this position. But they are very useful to understand and to get a better intuition of the overall regulatory logic of a system. Moreover, most of the data we have, at least for the systems we have been studying, are also qualitative. So I will show you in a minute some examples of the data we use to build these models. But well, these are some of the reasons why we have chosen these types of models. Okay, so these are some of the 2 very well chosen examples of the information we can translate into these rules. So imagine here we have roots of Arabidopsis thaliana, and we have a gene that can be, whose expression can be seen in blue. So here we have the wild type of the plant, and we can see the gene being expressed in blue. We have 2 single mutants, this one, and we see that the gene is still expressed as in the wild type. But we have a double mutant in which none of these elements are present, and neither is the blue, let's call it the blue gene. So we can translate this into an OR rule, in which if we have either of these elements or both of them, then we will have the blue gene being expressed, but if we don't have any of them, then the blue gene is gone. Yeah? And we have similar examples for the AND rule. I won't detail this one, but it's very similar. So this is more or less the kind of information that we find in the literature. Not only this, we also have access to databases, sometimes with more quantitative data. But this is the vast majority of the data that we have. So this is another reason why we have chosen this type of discrete model. So this approach has been used to study many systems in Arabidopsis thaliana, mainly in the group of Elena Alvarez-Buya, in which I studied the PhD, as Rafael was mentioning. They have studied the development of the flower organs in Arabidopsis. We also studied the cell-fate differentiation in the epidermis, which is the most external layer of the leaf and the root. Eugenio Speitea was studying also cell-fate determination in the root meristem, in this pool of undifferentiated cells. And maybe this image would help understand, or at least illustrate, how this system in many senses resemble the colon system that Philip Maine was talking about. There is this niche, and there is a set of undifferentiated cells, and then a transition and amplification zone, and then the differentiation and elongation zone over here. But I will be talking about this system in a little bit more detail, which I studied in collaboration with Jana Jatko in the Czech Republic at the Masaryk University. So there is a third type of plant meristem. Rafael was talking about the shoot meristem and the root meristem that give rise to the aerial and underground structures correspondingly, but there is another type of meristem, which is the vascular meristem, and this meristem gives rise to the vascular structures that help the plant transport nutrients, water, and signals along the plant. These vascular structures can change in their organization in the different parts of the plant, in leaves, in the shoot, in the basal shoot, in the root, but it always exhibits something common, some common pattern. It always has three types of cells, which is the procambium, which contains the undifferentiated and proliferating cells, the phloem, or the young phloem, which is here in purple, and the xylem, which is in green. And depending on the part of the plant we are looking at, these structures can be arranged slightly differently, but the procambium is always in the middle, as you can see here. So we decided to study how this pattern of procambium, phloem, and xylem emerges during the development of the plant. And we focused on the vascular bundles of the shoot. These are individual bundles, each of these ones. So this is a section of the shoot, and here is one bundle in which we can see the histological but also molecular differences between each type of cell. And we can trace the phloem cells, the procambial cells here in the middle, and then the xylem cells. Each cell type is characterized by a set of specific genes being expressed in them. And we use these as markers of the cell types. So in order to study this system, we put together all the available information up to 2012. And we integrated it into a network with different components. It includes mostly genes and transcription factors, but also some hormones and some small RNAs that can move between cells and couple the neighboring networks of the cells. So this network contains data for expression patterns, for loss of function mutants, overexpression mutants, protein-protein interactions, hormonal treatments, and other types of data. So we tried to test whether this network built on the available data was sufficient to explain or to reproduce the formation of these three regions in which the markers, these markers, are specifically expressed. We specified the rules. This type of specification is equivalent to the truth tables with zeros and ones that I was presenting before, but this, of course, is more synthetic. These are all based on available experimental data that had been at least once repeated. Then we specified the simplest spatial model that we could come up to. This model consists of three compartments, each corresponding to the position of procambial, phloem, and xylem cells. Each compartment has exactly the same network as the other, and has a possibility to communicate with the neighboring compartment with some rules that are also the same for all the three of them. The molecules that can move between cells are the ones that are indicated here in squares. This is a plant hormone, this is a small peptide, and this is a small RNA. The small peptide and the small RNA are known to move by diffusion, by some holes that are located in the plant cell walls, that are called plasmodesmata. And this hormone is known to move actively with some transporters. So in every time step, we update the logical rules according to this specification. And then we allow the mobile elements to move, some of them by diffusion, just by a discrete version of the Laplacian regular diffusion. And the auxin can move depending on the presence of the transporters, which are also part of the network. So when we modeled this, we realized that our network was not sufficient to recover or reproduce the specific expression patterns in each compartment. It could reproduce some important aspects of the system, but not some other very important ones. So we looked into data for the root and for genes of the same family, but very closely related genes to those in the network. And we chose these interactions in red as potential interactions that could be occurring here, and postulated them as part of the network. This is, of course, a very subjective way to introduce new interactions to a network. And we are now trying with David Rosenblut and other people to generate methods to try systematically all the possible single interactions that can be added to a network. Here we used, let's say, a conservation criteria to introduce these new interactions. So if these interactions were observed in other parts of the plant, or in sister or very close genes from the network, we introduce them. And when we do this, then we can, even if we start each compartment with exactly the same conditions, and which is not the case here, but even if we do that, the system attains different final attractors. One that has the markers for the xylem, one that has the markers for the procambium, and another one that has the markers for the phloem. And most importantly, well, at least for us, because we were very interested into looking at that, this system can also reproduce the concentration profiles, at least broadly, of these three plant hormones that were included in the network. And this depends mostly on the presence of these boundaries, which are zero-flux boundaries, and the initial presence of auxin. So, in another way to put it is that if we start the system with the same network and allow the hormone and the small peptide and the small RNA to move, then this is enough, and introduce the interactions that I told you about, then this is enough to attain three different network configurations that correspond to the cell types that have been observed. So from this, we thought that these interactions would be very good candidates to start testing in the laboratory for particular interactions, because when we introduce them, then we have a network, let's say, complete enough to reproduce the observed patterns. So we first tested the model by simulating some mutants, some mutants of the hormones, of the genes that produce the hormones, and also some mutants of the transcription factors, and they all matched very well the profiles, the gene expression profiles that have been reported for mutants. So we thought that, okay, we could then start testing some of these predictions, and the first one that we tested was this positive regulation coming from the cytokinin signaling pathway, which is a plant hormone, to a transcription factor, which is AAPL1. And some preliminary data showed that when we add cytokinin to the vascular tissues, here and here, then APL1, which is marked in blue, is more expressed. So this tells us that maybe these interactions that we were postulating as part of the network, by using our model to test the sufficiency of the interactions, were good candidates, and we are looking into more detail. Well, Jan in the laboratory in the Czech Republic is looking into more detail to these interactions. Then, because we are interested to, as I was telling in the beginning of the talk, in the conservation of processes, of developmental processes in different plants and eventually in different lineages, we started looking at this system, which is another plant model system. It's Cinia. And what has been seen in this plant is that an isolated cell of the leaf can be put in a solution of auxin, which is the plant hormone I was talking about. And it goes from being a regular leaf cell to being a procambial cell and eventually a xylem cell. So we tried to see if the network that we studied for Arabidopsis could reproduce this feature of Cinia behavior. And so we tested the model in a single cell version, not with the 3 compartments, with a single cell version, beginning with something similar to a solution of auxin. And we observed that indeed the system goes through something like procambial, and then it attains a xylem identity. What this tells us is that even if the components of the networks in Cinia and Arabidopsis are slightly different, because we know some of the components of the Cinia network, they are very similar, but they have some differences. And so even if there are these differences, the dynamic global behavior of these networks must be very similar, or at least this is what we think from these preliminary simulations. And now we are looking into more detail to the Cinia network and trying to do some comparative studies, but not only of the gene sequences or the protein shapes, but of the whole regulatory network underlying vascular development. And this is more or less the kind of thing that we would like to do when I talk about comparative studies in development. We would like to go beyond comparing sequences, gene sequences or anatomical structures alone, but we would like to compare the processes that give rise to these structures or patterns in this case. So some conclusions of this particular model is that we actually got a framework that enabled us to integrate available data and continuously emerging data from the lab of Jan. And this alone, I can tell you, is a very useful tool when you're working in a lab where lots of data are arising all the time. So this allows us to have a common framework to integrate and analyze this data. It also provides a possible mechanism that renders the patterns for the vascular bundles. It gives rise to new testable predictions that are being now tested experimentally, and that seem to be rather correct, at least one of them, the one that we started testing first. And well, it's a working model that helps us study or at least approach other systems in plant development with more intuition of what the overall mechanisms or vascular development are. Of course, the model has some important limitations, and the first one is that what we modeled as 3 compartments is actually should be actually a model of 3 populations of cells. And this is important because there are also subtypes of xylem and phloem cells, and if we eventually want to know how these subtypes are being specified, then we have to use a more population-like model with cells. And another important limitation of this model is that it can help us understand how these patterns are attained and how the vascular organs are specified, But it doesn't help us understand how these patterns can change, or this arrangement can change along the plant, the body plan of the plant. Also, these patterns change in response, as I was saying at the beginning of the talk. They change in response to environmental factors and stimuli. So we would like to include some of the signaling pathways that link environmental factors to this network, so that we can study the plasticity of this system in different environmental conditions. This plasticity, what we call phenotypic plasticity, is very important in ecological terms, and now it's being seen that it can also be important in evolutionary terms, as plasticity can be a source of variation, of important variation, that in sometimes can be inherited to further generations. Okay, and what I started to say just a few minutes ago is that these kind of models are opening new ways of linking development and evolution. So development and evolution have a long story of coming together and apart, and they came together, as I'd say, again in the late 80s in this Evo-Devo area. And now with these models, I think we have tools to make to study development in the context of these functional models that contain not only genes, but also hormones and maybe even environmental factors or physical and biophysical mechanisms. This also provides us with a tool to make comparative studies of the nature I was mentioning before. So comparative studies of models within and among lineages. And they help us to think or ideally postulate some structural or dynamical properties underlying the conservation or the innovation of shapes and patterns. This kind of approach to development also helps us think in terms of systemic properties that are often overlooked when we focus too closely on one or a few genes, such as modularity, robustness, and plasticity, which I mentioned briefly, and can help us integrate all the aspects of development that are now known to be important. So these images only aims to illustrate how the development of a very, very simple organ, like the root of Arabidopsis, depends on, yes, complex regulatory networks, but also environmental signals, the signaling pathways that link the organism with the environment, the formation of gradients. And as you can see in this, in the case of the vascular development and cell fate determination, these gradients are generated, most of the cases, at least in plants, by the cells themselves. This is also important in plants because plant cells cannot migrate. They have rigid cell walls that prevent them from moving. So unlike animal cells, plant cells remain in the same relative position. And what seems to be moving a lot are transcription factors and other molecules that can move through plasmodesmata, these holes in the cell walls that I was talking about. Okay. So this way of approaching development, focusing on developmental modules and processes, can help us, as I was mentioning, compare what's the process of the development of plants and animals, which have their whose common ancestor was a unicellular organism. What this means is that multicellular development was invented, let's say, twice independently in these lineages. So it is very useful to compare, I think, the processes of development in plants and animals so that we can postulate or find common aspects of these processes. Stuart Newman and Bad are trying to do this kind of comparison, of broad comparison. And in order to do that, they have proposed the term of dynamic patterning modules. So they define one of these modules, DPMs, as being conformed by molecules, but also by the physical and physicochemical processes that the interactions among these molecules enable. So in each of the patterning modules that they define, let's say here, there are a set or a family of molecules, but also physical principles that are being enabled by the interactions or the properties of these molecules. So these modules consist both of biochemical and biophysical components, and as it is mentioned here, they have some evo-devo role, some developmental role in animals. So for example, the module for addition can account for the multicellularity, for cells staying together, and relies on the presence of cadarins. This is a very simple one, but for example, they also define a module that can account for the formation of multiple cell types based on the physical principle of lateral inhibition, and often relies on the presence of notch and similar molecules. For example, for this particular module, they say that when we have this module acting on the basis of notch and its interactions with other molecules, it can exhibit lateral inhibition, and no matter where, in which part of the animal it acts, or at which point of the development, it will give rise to the sort of patterns we all know and we were talking about yesterday. But I think this is a very good example of this notion of modular development, as they give examples of how this lateral inhibition module can act with the same molecules in Drosophila, in zebrafish, in vertebrates, in the formation of hair follicles, and in many other developmental processes in animals, on the basis of the same molecules interacting. And so they define a set of these dynamical patterning modules and propose that the combination of these modules at different at different moments of development and in different parts of the animal can give rise to the basic characteristic of animal body plans. So here they have all the abbreviations for the patterning modules and illustrate how these modules acting in combination in different sequences can give rise to structures like this one, with different cell types, axis, polarity, and a complex structure in general. What we did with Valeria, Stuart Newman, and Carl Niklas was we tried to do the same exercise but for plants. So we postulated a set of dynamical patterning modules for plants, and here they are. I won't detail all of them, but as I was mentioning before, this diffusion patterning module is very important. It's based on the presence of these plasmodesmata that are formed after or during cell division, and that can give rise to cell differentiation. So there are all these 6 basic patterning modules that we defined, and we also postulate some sequences on which these modules could be acting in combination, giving rise to the basic structures of plants. And the vascular determination and patterning system that I was talking about just a few minutes ago is a particular example of a combination of some of these patterning modules. So this is one, let's say, theoretical or proposal that we have to integrate gene regulatory networks into a larger framework that includes physicochemical and other types of interactions that take place during plant and animal development. We then traced the origin of the molecules associated to each of these modules to the plant phylogeny, and discussed the origin of these patterning modules in plants, in the first plants. So, well, just to finish, I would like to end with this very basic or general schematic representation of what we would like to do eventually. So, in order to understand plant development, how plant shapes and forms arise, but also how they change in the lifetime of an organism and during evolution, well, we have to study the processes of plastic cell determination and patterning. We can do it by using network models, but not only. But also the relation of these processes with evolutionary processes that underlie the fixation of patterns in plant lineages, and with ecological processes that underlie the interaction between organism and environment, and that we have seen are also very, very important in the plasticity of plant, but also animal development. So I finish with that, and I thank you for your attention. Here are some of the references that you can consult if you want to read more about this. Thank you.